A good morning or afternoon to you. Thank you for joining today's webinar. I'm Jan Reichert, Executive Director of the Antibody Society, and I will be your moderator. This webinar is the sixth in a series of webinars designed to inform and educate our members as well as the broader scientific community about antibody discovery and development. The title of today's webinar is Antibody FC Engineering, Designing Antibodies for Cancer, COVID-19, and Beyond. Our speaker is Dr. Alicia Chenoweth of King's College London. This webinar is being recorded. We'll take questions at the end of the presentation, so please do add your questions to the Q&A box in the viewer. Without further ado, I now turn the show over to Dr. Chenoweth.
Thank you, Jan, and to the Antibody Society for the opportunity to give this seminar today. And thank you to all of you taking the time out of your day to listen to FC Engineering. So, monoclonal antibodies are a very successful form of biotherapeutic that have revolutionized the treatment of a range of previously incurable or chronic diseases. The wide applicability and efficacy of monoclonal antibodies as a therapeutic has translated into great commercial success, with 5 of the top 10 best-selling drugs in 2020 being monoclonal antibodies, and the global antibody market accounting for over $100 billion. The development of monoclonals is growing every year, with great numbers now being explored in clinical trials. The success of monoclonal antibodies as therapeutics stems from their ability to be easily adapted for many different types of diseases, from cancer to autoimmunity to infectious diseases. Usually, if there's a target, a monoclonal antibody can be developed. They are very specific for this target and very well tolerated in the body, as they are naturally produced in response to pathogens, which leads to less off-target side effects. The binding of the target is mediated by the Fab portion of the antibody, the top part, which is the prongs of the Y shape. The rest of the antibody, the FC region, which is the single stick part of the Y, is a very constant and conserved region, which interacts with the FC receptors on the surface of immune cells and can induce very potent effector functions. Antibodies are multifunctional and can induce different effector functions depending on what you want to do for the disease you're trying to target. The very basic and simple function of an antibody is binding to the target and blocking its interaction with another protein, such as binding to a cytokine or cytokine receptor and blocking that interaction, or binding to an infectious disease particle such as a virus, and blocking the binding of the virus to the host cell. This action at the base level only requires the Fab portion of the antibody and doesn't really need any immune cell interaction or FC-mediated effector functions. Contrastingly, functions like antibody-dependent cellular cytotoxicity, phagocytosis, degranulation, all of these potent pro-inflammatory cellular functions require not only the Fab region of the antibody to bind to the target antigen, but also the FC portion of the antibody to bind to FC receptors on the surface of immune cells. An example of this is the Fab portion of an antibody binding to a receptor expressed on cancer cells, like HER2 in breast cancer, and the FC portion binding to FC receptors on NK cells to induce ADCC to kill the cancer cell. However, not all FC receptors on immune cells induce pro-inflammatory signaling. In fact, they can induce inhibitory signaling and actively block or dampen the activating signaling induced by other FC receptors or the B cell receptor, for example. This may be the desired effect for a therapeutic to dampen or block the activating signal of the BCR on B cells. Like the pro-inflammatory signaling, this effect requires both the Fab region of the antibody to bind to the target and the FC portion to bind to the inhibitory FC receptor on the surface of the cell. Another potential function of antibodies is agonism. So that's where the antibody binds to a target receptor on the surface of cells and clusters those receptors to induce signaling. This sort of signaling could be co-stimulatory molecules, such as CD28 on T cells, or to induce apoptosis of the target cell by clustering death receptors. This function, as I'll explain a bit more later, doesn't seem like it would actually require the FC portion, but it can, in fact, play quite an important role. So we can see for many of the therapeutic functions desired for monoclonal antibodies, the FC region of the antibody can play a large, crucial role. Thus, selecting and optimizing the FC domain of the antibody is a really important step in developing the most effective therapeutic for your disease of choice. So one of the first decisions to make is which antibody class to base your therapeutic antibody on. There are 5 different classes of antibodies naturally produced in humans, all with different functions and properties. They can have different structures, ranging from monomeric individual proteins or to big hexameric structures, different ability to activate complement, different serum half-lives, and differing ability to induce immune cell activation. Historically, IgG has been the antibody class of choice for developing therapeutics due to its wide range of pro-inflammatory effector functions, complement activation, and long serum half-life. However, more recently, other antibody classes have been started to be investigated as potential therapeutics to harness their more unique characteristics for specific applications. For example, IgE is an antibody class commonly associated with very pro-inflammatory allergic diseases or protection against parasites. A drawback of IgE is that it does have a quite short serum half-life, around 2 to 3 days, compared to IgG's 21 days on average, but has a very high affinity for its Fc epsilon receptor, expressed on many cells such as mast cells, basophils, monocytes, and macrophages, which not only contribute to having a long tissue half-life, but also to the very pro-inflammatory effector functions it can induce through these cells. There's also no known inhibitory receptor for IgE, unlike IgG, which can also contribute to its strong pro-inflammatory response. So in a cancer setting, for example, inducing a very pro-inflammatory environment is actually quite an advantage. To this end, IgE-based therapeutics are now in phase 1 clinical trials after in vitro and in vivo studies demonstrated a greater solid tumor killing ability of IgE therapeutics versus IgG. A concern about these IgE therapeutics was that they may induce an anaphylactic or allergic type response, but so far these antibodies have actually been well tolerated and quite safe. IgM as well is a unique antibody class in that it forms pentamers and hexamers. Because of this, it has many more antigen binding sites compared with IgG, which allows stronger avidity type binding to targets. This is definitely a benefit in low expression targets or difficult targets, and why many of the IgM-based therapeutics that have been tested in clinical trials have been against poorly immunogenic targets, such as lipopolysaccharides, glycolipids, and glycans. Although it doesn't have quite as strong cell-based pro-inflammatory effects as IgG, IgM is able to very potently activate the complement pathway, which can kill the target cells without the need for immune cells. And the large size of IgM structures also allowed for multiple target binding in the same structure, such as different epitopes of a viral particle, which can lead to greater neutralization of the virus. There's a couple of IgM-based therapeutics in clinical trials, mostly for cancer targets at the moment, but recent studies have shown that IgM-based antibodies have better neutralization potential compared to IgG against Covid-19 and influenza B, so it seems like there's a potential for IgM therapeutics to move into the viral infection space. IgA is a uniquely structured antibody as it has the ability to form both monomers and dimers. There's no IgA-based therapeutics currently in clinical trials, but in vivo and in vitro studies have suggested it may have more potent anti-cancer activity compared to IgG, especially by neutrophils. However, they've been a bit trickier to investigate due to mice not expressing the IgA receptor, and thus preclinical models have been a bit harder to find out than IgG. And the antibodies do have a bit more aggregation potential and does have a shorter serum half-life. However, there have been many advances in the engineering of these IgAs to overcome these difficulties, which may allow in the future for them to be more explored in depth as a potential therapeutic. And finally, IgD has not really been investigated for therapeutic use so far, as so far we haven't seemed to find many beneficial properties compared to the other subclasses. So it has a short serum half-life, doesn't induce much immune cell activation or complement, so as of yet doesn't seem to be an obvious use for it as a therapeutic compared to IgG. So I've given you a bit of an overview of the different antibody classes and their benefits, but I'll be focusing from now on IgG specifically, as they are the bulk of the therapeutic antibodies developed and used. So IgG has 4 different subtypes with different properties and effector functions. For those antibodies that require the Fc portion, if you want to induce ADCC or phagocytosis, for example, the traditional choice of antibody has been usually an IgG1. So it binds to all the Fc gamma receptors on immune cells, it's got a long half-life, binds complement, it's a very pro-activating antibody, basically. For those antibodies that don't really require the Fc portion, for example, if you want to use a blocking antibody, usually an IgG2 or IgG4 subclass would be used, as they have a much more limited binding to the Fc gamma receptors, and they don't bind as many immune cells and don't induce effector functions. And they also have quite low to no complement binding, but they do also have the same serum half-life. So they were seen to be better if you wanted a less activating antibody. And IgG3 is not really used so far as a therapeutic. It has very similar effector functions to IgG1, but it has a lower half-life, potentially due to its longer hinge region, which is more susceptible to proteases, and is much more variable in different allotypes, so is a bit more difficult to work with than IgG1. So the majority of therapeutics in clinic are either on an IgG1 backbone, and there are some IgG2 or IgG4 if no Fc-based effector functions are required. As I'll talk about later, however, using these native IgG wild-type backbones may not be optimal for therapeutic function. And recently, through better understanding of the structure and function of these antibody subclasses and how they interact with their receptors to induce effector functions, we are finding that specific targeted engineering of the Fc portion of these antibodies can actually improve the therapeutic effects, and we can tailor them to more specifically, more effectively, and more safely induce the desired function for the disease of choice. So firstly, I'll be talking about how Fc engineering is being used to improve blocking antibodies. Although these antibodies don't require the Fc region for their blocking activity, we can still use the Fc engineering techniques to make these safer and more effective. Firstly, by simply removing the Fc effector functions, which are not required, and we can improve the safety of the antibodies this way. For example, many of the antibodies that are currently in clinical trials for COVID-19 treatment have had their Fc regions engineered to remove all effector functions to avoid potential antibody-dependent enhancement. ADE has been demonstrated in previous viruses such as dengue, influenza, HIV, and Ebola, and it does so in kind of two main facets. Firstly, internalization of the virus by Fc receptors on the surface of the cell can lead to infection of that cell and further production of virions. Or secondly, increased inflammation induced by Fc receptors through immune cells in the airway, for example, can lead to more severe disease symptoms. Removal of the Fc effector functions may mitigate these effects. It's not yet been shown that AD would be a problem in COVID, and in fact, it might not be. But original development of some of the COVID-19 antibodies have taken the safest approach and removed the effector functions just in case. Another case in which you'd want to remove the effector functions is when the target's on immune cells, for checkpoint inhibitors, for example, as you don't actually want to kill these cells. So how do we remove effector functions? As I mentioned before, the classic way was to change the subclass of the antibody from an IgG1 to an IgG2 or 4, which have more limited Fc receptor interaction, and thus has been seen to be less activating. There are many antibodies used in clinic that use this strategy very effectively, such as the anti-PD-1 checkpoint inhibitors and anti-CTLA-4 checkpoint inhibitors. However, although IgG2 and IgG4 have a more limited Fc receptor profile, they do still bind to some Fc receptors and can induce effector functions, as demonstrated in vitro by an IgG4 anti-PD-1 antibody actually inducing activation of the receptor instead of the blocking activity that was required. And this was because it still had Fc effector functions. In addition, the use of an IgG4 backbone on an anti-CD28 antibody induced clustering and signaling of the receptor instead of blocking the receptor, causing severe cytokine storm in healthy volunteers in a phase 1 clinical trial, through a scaffolding action by the Fc portion of the IgG4 binding to an Fc receptor. So this approach, while having found some success in the clinic, isn't perfect. Another way to remove effector functions from an IgG is to remove the glycan, which is attached to the Fc portion, often by mutating the asparagine residue it is attached to alanine or glutamine. This stops binding of the antibody to all Fc receptors and has been used in clinic for anti-PD-L1 antibody, as well as a recently approved anti-CGRP antibody for migraines. However, this approach is quite a drastic alteration to the structure of the antibody and may also alter the pharmacokinetics of the antibody, such as by reducing its thermostability, and then therefore it may not also be a perfect solution for every situation. Finally, removal of effector functions can be achieved by mutation of the Fc region, which is becoming the favored way to achieve this. This could be by creating hybrid antibodies, which combine parts of IgG1, IgG2, and/or IgG4 to create an antibody that doesn't bind any Fc gamma receptors, unlike the individual wild-type antibodies themselves. Some examples of this include having an IgG1 backbone, but substituting the lower hinge and the Fg loop, which are two quite important regions of contact of the antibody with the Fc receptors, for the sequences in IgG4. And this is used in durvalumab, an anti-PD-1 antibody, which is currently in clinic. Another example is another anti-PD-1 antibody in phase 3 clinical trials, which has an IgG4 backbone, which, along with a couple other mutations, has the lower hinge region of IgG2 substituted. And some of the COVID antibodies in development are also using this approach to remove effector functions. Another way is to point mutate key regions of the antibodies which interact with the Fc receptors. As I mentioned, the hinge region is a very important region in Fc receptor binding, and mutating two residues in this region of IgG1 to alanine, known as the LALA mutation, decreases binding to receptors by over 100-fold. This technique has been successfully used in 2 approved antibodies so far, and although used successfully and it does decrease binding significantly, it does not remove binding to all Fc receptors. And there are still ongoing investigations into finding the best and most effective way of removing all the Fc receptor binding without compromising structural integrity and pharmacokinetics. And to this end, there are many new mutation combinations being discovered all the time. Other than removing effector functions, another way we can improve the efficacy of blocking antibodies and antibodies with other effector functions is to increase their serum half-life. IgG usually has a half-life of around 3 weeks, which is due to its ability to bind to the receptor called FcRn. Very basically, when antibodies are uptaken by penocytosis into an endosome of a cell, they bind to FcRn in these low pH conditions, around pH 5 to 6.5. If they are bound, they get recycled and released back into the serum. If they don't bind, they get degraded. So by engineering the antibody to have increased binding to FcRn, we can increase the half-life of these antibodies up to almost 3 months, as they will continue getting recycled rather than degraded, which can increase efficacy of the antibody and decrease dosing frequency, which would result in a lower overall treatment cost. 2 common ways this is done is by the LS mutation, also known as extend, which is 2 amino acid mutations, and the YTE mutation, a triple amino acid mutation, which also decreases effector functions. The LS mutation has an 11-fold higher binding affinity to FcRn at pH 6, which translated to an approximate 52-day half-life for the approved anti-CD5A antibody, compared to eculizumab, which is basically the same antibody, same target epitope, just without the half-life extension. So it went from about 3 weeks to 52 days. And because of this, the dosing schedule could be increased from every 2 weeks to every 8 weeks and have the same efficacy for the patients. So similarly, the YTE mutation drastically increases half-life, in some cases over 100 days. Interesting, however, this mutation also decreases the Fc-mediated effector functions of the antibodies, such as ADCC. So it's best used for antibodies which don't require Fc-based effector functions, such if you just want pure blocking against viral diseases. Increasing the half-life of antibodies against infectious diseases like COVID mean that it could potentially be used as a prophylactic strategy for high-risk groups. For example, immunocompromised patients or people that have suboptimal vaccination responses, which could potentially protect them up to 3 months per dose. So in some cases, rather than decreasing effector function, we actually want to have increased pro-inflammatory functions such as ADCC and phagocytosis. So how do we make this happen? As I mentioned earlier, IgG has both activating receptors and an inhibitory receptor, which is able to shut down the signals of the activating receptors. So there's always this balance between activating and inhibition. To improve effector functions, we want to skew this balance more towards activating and away from inhibition. So much work has gone into understanding how these antibodies bind to these different receptors, both activating and inhibitory, and the differences between them. And from numerous crystallography and mutagenesis studies, key residues and regions for binding to the different receptors have been identified. Once the regions and residues were identified, we could then start modifying them to try and achieve the activating profile of choice. So the most common strategy is to increase binding to one specific activating receptor, Fc gamma R3A, which is a very potent inducer of ADCC and pro-inflammatory effector functions on NK cells and macrophages. So one way this can be achieved is through the mutation of 2 or 3 residues, the S239D, I332E, and or A330L, which increases binding to Fc gamma R3A and thus increases NK cell-mediated ADCC. As you can see on the top right, in an anti-HER2 model by Lazar et al., the double mutation in blue and the triple mutation in brown were able to induce significantly more ADCC against HER2-expressing cell lines compared to wild-type IgG1. The double mutation has subsequently gone forward and been used in an approved antibody for non-Hodgkin lymphoma therapy. The double mutation tends to be used a bit more, as though it does induce slightly less ADCC compared to the triple mutation. It does retain complement-mediated killing, unlike the triple mutant, which can be another mechanism by which the antibody works to kill the target. A similar strategy has used 5 amino acid mutations to not only increase Fc gamma R3A binding, but also decrease binding to the inhibitory receptor, thus skewing the balance of activating inhibitory even more. As you can see on the bottom right, these are biocode graphs from Nordstrom et al., which demonstrate that these antibodies containing these 5 mutations in the red binds much better to Fc gamma R3A and has decreased binding to Fc gamma R2B compared to wild-type IgG1 in the blue. And it also retains binding to FcRn, so retains its half-life. These mutations were incorporated into trastuzumab, the anti-HER2 antibody against breast cancer, to make margetuximab, which in its phase 3 trial was able to more effectively increase progression-free survival in patients compared to trastuzumab. And this is the same antibody, just without these 5 mutations. So you can see that skewing this balance of activating versus inhibitory can actually lead to more effective therapies against cancer, even with the same target. Another strategy to improve cell-based effector functions is to try and improve antibody-dependent cellular phagocytosis. As often in a tumor microenvironment, there are quite few effector cells such as NK cells, but a lot of macrophages. The mutation G236A was used to selectively increase binding to Fc gamma R2A, a receptor that's abundantly found on most immune cells, including macrophages, and does mediate phagocytosis. This mutation alone, however, does decrease affinity to Fc gamma R1, which is another activating receptor which can also mediate phagocytosis. So to combat this, this mutation was combined with I332E alone, or the S239D I332 mutation, which can restore Fc gamma R1 binding. And as I mentioned earlier, also increase Fc gamma R3A binding. So as we can see on the right, studies by Richards et al. Demonstrated that addition of these mutations with the G236A induces much greater ADCP phagocytosis by macrophages compared to wild-type IgG1. And these mutations are just the tip of the iceberg as well. As we learn more about the interactions between the different Fc receptors with IgG, as well as the development of faster, high-throughput mutational screens, we're finding novel mutations to selectively modify the activation R profiles of the antibodies to induce the best result. Another common way to increase binding to Fc gamma R3A, the NK cell pro-inflammatory receptor, for greater ADCC induction is to modify the glycan in the Fc region. As I mentioned earlier, removing the glycan ablates all effector function. However, if you move just a single residue, the fucose residue on this glycan, it increases affinity to Fc gamma R3A, but not to the inhibitory receptor. So once again, you get this skewing away from inhibition and towards activation. This has been quite a popular strategy to use in many cancer antibodies. An example of this is obinutuzumab, which nearly doubles progression-free survival in chronic lymphocytic lymphoma patients compared to rituximab. And both antibodies are anti-CD20, so against the same target, but obinutuzumab has no fucose residue. So removal of fucose has not only been looked at for cancer targets. Elite controllers of HIV infection naturally produce antibodies with low fucose, indicating a better control of the virus when these antibodies are more activatory. However, this is not the case with dengue, when in fact low fucose antibodies have been associated with enhanced pathology, likely due to antibody-dependent enhancement. So obviously every virus is quite different into what response it needs. And so over the last year or so, many people have been wondering, how does COVID respond to these antibodies? And is this a viable strategy to increase effectiveness of monoclonals against SARS-CoV-2? A recent study demonstrated that there's an increase in afucosylated antibodies in critically ill patients compared to those with mild symptoms, and postulated that this could be due to the afucosylated antibodies increasing the inflammation and exacerbating the infection. So potentially for therapeutic COVID antibodies, increased effector function by removing the fucose may not be ideal, although it is a very viable strategy against many other diseases. So really it's all about knowing what disease you are targeting and how best to attack it, and then using these FC modification tools to help. So we've covered removing effector functions and increasing activating functions. Another really interesting field of FC engineering is engineering antibodies to have increased binding to the inhibitory receptor, Fc gamma R2b, and how this is being used for different types of therapies. So Fc gamma R2b can inhibit the activating signals made by other Fc receptors, as well as the B cell receptor, where it's co-expressed on B cells. So the obvious reason to increase affinity to Fc gamma R2b, the inhibitory receptor, is where you have too much inflammation and want to suppress it, such as in autoimmunity or allergy. As I mentioned before, many studies have gone into investigating how the antibodies bind to the different Fc receptors. And in doing this, residues which increase binding to the inhibitory receptor Fc gamma R2b have been discovered, such as the self mutants, which increase binding to Fc gamma R2b by over 400-fold. These mutations are being used in clinical trials. One antibody that uses them is oboleximab, which is an anti-CD19 antibody designed to suppress B-cell signaling and proliferation in autoimmune diseases such as lupus. In an allergy setting, there's an antibody called omalizumab, which targets the IgE and has been quite effective in clinic. However, not for all patients and not completely. In an effort to try and increase efficacy, these self-mutations have been added to it to create a new antibody called XmAb7195, which demonstrates greater IgE removal and can also inhibit IgE production by B cells for the treatment of allergy. One potential drawback to the self mutations is that due to the high similarity between the inhibitory receptor Fc gamma R2b and one allele of an activating receptor Fc gamma R2a, which is expressed in some people, the self mutations also increase affinity to this activating receptor, which obviously could cause some unwanted activation effects. So to combat this, another mutation set that has been used is called V12 and incorporates 6 point mutations. And this also increases Fc gamma R2b in affinity, but it doesn't increase binding to any of the activating receptors, which possibly might be a better and safer way forward for this engineering strategy. As I mentioned before, antibodies can also have an agonist function. That is, they can bind to a receptor on a cell surface, aggregate them, and induce signaling, such as T-cell activation or apoptosis through death receptors. For this agonistic function, signaling and effector functions through the Fc receptors aren't required. However, it doesn't mean they can't play a role. In fact, Fc gamma R2b in particular is quite effective as a scaffold. That is, the Fc portions can bind to the Fc gamma R2b on a different cell, but rather than induce signaling through those receptors, it just kind of helps to cluster the target receptors more effectively. So once this indirect function of Fc gamma R2b was discovered, people started to wonder whether increasing the affinity for Fc gamma R2b was able to further increase the ability of this receptor to scaffold and cluster the target effector receptors. And it does. So antibodies that have been developed to have increased Fc gamma R2b binding, the ones I just showed you now, such as the self, can induce more apoptosis and decrease tumor growth, or more agonism than an unmodified IgG1. So it's just a really interesting way that Fc engineering can improve the function of an antibody in which Fc function itself isn't required, but it can be very useful. So the majority of the techniques I've shown you for Fc engineering have been relatively direct, to remove or improve effector function by directly modifying how they interact with the Fc receptors that induce them. A really interesting field is sort of indirectly modifying the antibody for increased function by changing its flexibility and structure, which doesn't directly affect binding affinity, but it does affect function. So an example for this is once again for agonism. So in the context of enhancing affinity to Fc gamma R2b, it does rely on the need for that receptor to be present at the site of agonism. So it is really limited by the availability of Fc gamma R2b presence. An alternative approach that's being investigated is to create agonists that don't require Fc function or Fc binding to any receptor to induce a similarly strong agonistic signal. One of the properties that sets the different IgG subclasses apart is the hinge region. And IgG2 has a really unique hinge configuration, which allows it to form two different structures. IgG2A, which is actually quite flexible and lets the Fab arms move around a bit more. And then IgG2B, which is a bit more compact and less flexible. These transition naturally between IgG2A to intermediate to IgG2B, but you can actually force the IgG2 into a structure into an IgG2B structure by mutation of a couple of the cysteine residues. What was quite interesting was that in a study of anti-CD40 agonism by White et al., IgG2 was found to induce much stronger agonistic signals than IgG1 in the absence of any Fc binding to anything. And specifically, it was the IgG2B structure that greatly induced agonism. So despite no Fc functions really needed at all here, it's all Fc independent, modifying the Fc region and hinge may actually provide an indirect way to make these antibodies more effective by altering the structure and the flexibility. So in summary, although unmodified monoclonal antibodies have had much success in the clinic against a range of diseases, there's still a lot of room for improvement for many of these antibodies. The work done in crystallography and mutation of these antibodies has allowed for understanding of the structure-function relationship of antibodies and their receptors, which has developed into being to fine-tune these antibodies for greater efficacy, safety, and specificity of the function that you want in a specific disease. The benefits of Fc engineering is that what you learn in one disease can easily be applied to other diseases. So antibodies that have been developed with increased Fc gamma R2b function for allergy are now being repurposed for agonistic antibodies in cancer. It's the same mutations, but a different target, different disease. And this allows for improved therapeutics for emerging diseases. We can use what we learned in other diseases right away and put them in a different therapy. And this field is expanding all the time. It's very exciting. The collaboration between different experts in different fields is really crucial. The antibody experts that understand the structure function of the antibodies and know how to modify them for optimal function and make them safer, and creating all these new improved modifications all the time, discovering really interesting features which can be exploited for therapeutic function. But you also need to know what the disease needs. So we need the disease experts who find a target, know what function is required for the best treatment, so that the antibodies can be designed accordingly. So I've given you a really brief look into the world of Fc engineering today. There's so much more that's being done, from looking at antibodies from different species like camelids to discover novel structures which may have more favorable properties that we can import, to creation of bispecifics, antibody-drug conjugates through modification of the Fc region, making new fragments, structures, fusions, hybrids. The field's really, really exciting. So I would just like to say thank you for all of you to listen today. Here are my socials if you forget to ask a question or you think of one later. Also, I just wanted to point out that my colleague Sylvia Crescioli and I have worked with Jan to produce a really great table with all the approved antibodies and all the information of the Fc modifications and why they were made, the backbones of the antibodies, which cell lines they were produced into, if they were fragments, antibody-drug conjugates, how they were conjugated into what. So it's a really cool table. So yeah, check that out, and thank you. I'll take some questions now. Thanks for listening.
Excellent. Thank you very much, Alicia. That was very comprehensive and thorough coverage of all sorts of antibody engineering techniques. And we do have some questions. So for the first question, it's actually two-part. How has glycoengineering of the Fc region been approached during the synthesis stage? Follow-on question. Are there preferred expression models that can be engineered for specific glycosylation patterns?
Yeah, sure. This is a really good question. So for modifying the glycan to remove the fucose, for example, there's, I guess, kind of two main ways it can be done, which is with kind of enzymes, which you can produce, add into while you're producing the antibodies. Or there are also specific cell lines that you can now produce antibodies in that naturally lack that kind of enzyme that will put the fucose on, so it will create naturally low fucose or no fucose antibodies. So if in the table as well, because there's a couple of afucosylated antibodies that have been approved in the table, if you look at the cell lines, some of those that have been made have been in cell lines that naturally lack the fucose enzyme. So that's how they made it, and some have made it a different way by putting enzymes in to prevent fucose. So yeah, there's also, obviously people have been looking at all the different other residues of the glycan, and that's done also very similar ways by just stripping it with different enzymes and all that sort of thing. So it's, yeah, cell lines and enzymes, pretty much.
Thank you.
Thanks for the question.
A follow-on, really easy question, but since you mentioned the table, one of the questions is, where can we find the table you mentioned?
Sure. So that's it's on the Antibody Society website. If you go into web resources and then hover over the antibody therapeutics approved or in regulatory approved, and then there'll be a little pop-up that says antibody therapeutics product data. Click that and then you'll find it. You can download it as well. Should soon. I think we're trying to get that functionality put in. So soon you'll be able to download it as an Excel. You can look at it and it's searchable. So if you're looking for just, say, fragments or ADCs, you can search the table and it'll come up with just those. So it's quite nice. Yeah.
Next question. What is your opinion on optimal FC design for bispecific antibodies where each target may optimally require different isotypes? For example, an anti-HER2 and anti-PD-1 bispecific antibody.
There's a really, really interesting question. So I guess I've not done too much work on bispecifics. I know that you can, I guess, modify each side a bit differently. So on the anti-HER2 side, you can make it kind of pro-inflammatory. On the other side, you can make it less. I guess, yeah, it would really depend on the function and what you're looking for. And I would have to do a lot more research on that because it's a really fascinating way. Because, yeah, if you want to have a pro-inflammatory anti-HER2, but you want to have no effector function and not kill off the PD-1 expressing cells, it is quite a tricky dilemma. Yeah, I really want to have a look that up now. It's quite interesting. So I can have a look and we can talk about it later if you like. I've not worked with bispecifics personally, but it's a really, really interesting question.
Two people have a very similar question. It has to do with, you know, if much work has been done on looking at the effects of the FC modifications on anti-drug response, so therefore the immunogenicity potential of the FC amino acid modifications.
Sure. So for a lot of these ones that I've shown today, that has been looked at because obviously they've been in clinic and reviewed, and there hasn't really been much. Usually, I think once you start to get over maybe 6 or 7, 8 kind of amino acid changes, then things start to look a bit different, and the anti-drug responses do start to come up a bit more. So most people working with engineering try to keep it to a smaller amount of modifications. Kind of where the hybrid antibodies come in work quite well because you're taking stuff that's already seen in the body. So IgG1 with a bit of IgG2 usually won't inflict much of a response, but you need quite a bit of modification, I found, usually to induce the anti-drug responses. And for the chimeric antibodies used for so long, they had a lot of mouse section on them and didn't induce much. So 1 or 2 amino acids, 3 or 4 here and there, have not so far seen to induce that much ADE. Doesn't induce, yeah, it still kind of looks like an antibody. Yeah.
Next question. What about the Fc gamma R3b enhancing mutations effect on Fc gamma R2c?
Right. So yes. So I'd imagine you would be talking about Fc gamma R2b on 2c, which is, so for those who aren't as well off understanding of the Fc receptor biology, Fc gamma R2c is an activating receptor which is expressed in about 20% of people, and it has the kind of extracellular domain of Fc gamma R2b, the inhibitory receptor, but the activating domain of Fc gamma R2a. So it binds pretty much identically as Fc gamma R2b to an antibody. So yes, with those situations, if you are trying to enhance Fc gamma R2b affinity, you will enhance Fc gamma R2c affinity. So for those, I guess for those patients that you're kind of worried about that, you may have to screen for Fc gamma R2c expression and see how, if it does induce quite an activating effect through that receptor. It's not expressed on too many cells and not very highly, as far as I'm aware. And as I said, only 20% of people, but it is definitely a consideration when designing these sort of antibodies against Fc gamma R2b. Yeah.
Next question asks for your opinion regarding do cyno monkeys continue to be appropriate models for preclinical studies of these Fc modified antibodies?
Yes, I did a lot of work in my PhD on this and comparing basically macaque and cynomolgus monkey Fc receptors and antibodies to humans. I guess the short answer is it's kind of as good as we've got so far, but the long answer is it's not perfect. It's really not. Like, the Fc receptors of cynomolgus monkeys are not identical to humans. And do bind different antibodies differently, especially once you start Fc modifying them. They do have quite different responses. So it's not a perfect system, and you really do have to take that into account. But it's a very tricky one because no model system is going to be perfect compared to humans. And I would suggest, if you're worried or you're thinking about it, just with your Fc modified antibodies, see how it binds to the different Fc receptors of cynomolgus monkeys and macaques to see if the profile is very similar to humans, because if it is, then they should be an appropriate model. But if it's not, then that's really something you're going to have to take into account because it can be quite different.
Thanks very much. Next question, in terms of prolonging half-life, what is your opinion on the use of histidine switch motifs to modulate FcR binding?
That's a really interesting question. It's something I have not looked at too much. I don't know all that much about it so far. I don't know too much. I'm happy to go have a look at it, and we can have a look afterwards. Sorry.
And my apologies to the long-suffering attendee who's been waiting for me to get to their question. I did not forget you, I promise. The question is around Fc engineering to improve safety of antiviral antibodies.
Mm-hmm. So yes, as I mentioned, for some viruses, a lot of people are taking kind of the safest approach, which is to eliminate all Fc-based functions, as a lot of the times the antibodies are kind of just used as a simple blocking, so just want to block the viral particle from binding to the receptor to the cell. It is, there is the worry about ADE, and that's why they have taken that step to kind of prevent any antibody-dependent enhancement to make the symptoms worse. As I mentioned, that's not always the case for all viruses, so it really depends on the virus itself. And so not all, I can't say in a whole, every single virus is going to react the same way to all the antibodies. So in some cases, to be safest, they've removed all effector functions, but for some viruses, actually, they do benefit from effector functions such as HIV. I guess the easiest way is, if you're not unsure, remove effector functions, but figure out whether the virus, how the virus pathology works, and what would be the best approach for it.
Thanks very much. Next question. Has it been confirmed on the protein level that Fc gamma R2C is really expressed in a significant manner?
I think so for certain cell types. So it's not as abundantly expressed as, say, Fc gamma R2A or Fc gamma R2B. But on certain cell types, I believe it has been shown to be fairly well expressed in the people that do express it, that do have the gene polymorphism to have Fc gamma R2B expressed. It is in some cells expressed quite significantly. Yeah.
And one person just wants to add a comment regarding the fact that IgG3, because you had mentioned that it's used in BioWA's Compligent technology, which represents IgG1, IgG3 Fc fusion, designed to achieve enhanced ADC. So I don't know if you have any additional comments to add to that comment.
Yeah, so I have started to read that IgG3. So when I mentioned that IgG3 itself is not used, I kind of meant as in a very basic way that the backbone as a whole, a wild type backbone, is not used. But there are, as this person said, amazing strides being taken into making these hybrid antibodies and fusion antibodies, which take kind of beneficial features of IgG3, because there are quite a number of them, and incorporating them into, say, an IgG1 to have better enhanced ADC, all that. So it's a very cool field, yeah. So yeah, sorry, I didn't mean to say IgG3 is terrible. It's not what I meant. But yeah, it's very exciting.
Well, with that, I believe we've come to the end of the questions. So in concluding, I'd like to thank Dr. Chenoweth for sharing her wealth of knowledge about Fc engineering and for the fantastic work that she and her colleague, Dr. Silvia Criscioli, did on the searchable table of approved antibody therapeutics, as well as those in regulatory review. Those are included also. So this table, as Alicia mentioned, it can be found in the web resources section of the Antibody Society's website. It does include a tremendous amount of new information on the format, Fc engineering, as well as the cell line expression system. So please do go and have a look at that. I also want to thank you very much for joining the webinar today. An on-demand version will be available in a few days. I'll send a link to it via email to everyone who registered. Please do feel free to watch this and any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.